All right, so once again, good morning, everyone. Welcome to the Adirondack Park Invasive Plant Program Spring Partner Meeting. This is always just fun to have a chance to see everybody. I've already met a lot of new people today, and then also many familiar faces. Those of you who know, I often start these meetings with a bit of a topical reading. So today I'm reading from Tom Kalinowski's Adirondack Almanac, and it's a fun little story. It's about amphibians. So towards the end of April, it seems that every day adds a new sound to the many natural noises that fill the air. And while many of these are made by birds, a few are produced by amphibians. So in the Adirondacks, the wood frog is normally the first of these moist-skinned creatures to begin its mating serenade. And shortly after the ground thaws in mid-April, it emerges from its state of hibernation and heads to the closest pool of water. Unlike other frogs that spend their winter underwater, these little brown creatures with their nice black masks spend the winter burrowed underground. So soon after they arrive at these vernal pools, they produce a series of quacking sounds. Many of us are quite familiar with those, but you're lucky if you hear them, because the wood frog's breeding season only lasts for a couple of weeks, and then they're done, and they go back, and they burrow back under the ground, and we don't get to hear them or see them again for another year. So even though the wood frog is out there only for a week or two, we all know the spring peepers come out for quite a long time. And one of the cool things about the spring peepers is because they're quite full-blooded, they adapt their calling to the temperatures. So on those mild spring nights, you can go out and it's just a cacophony, sometimes almost deafening. But on a frosty night like last night, or a frosty morning like this morning, we probably didn't hear much about peepers. So like the wood frog, the spring peeper spends its winter underground, burrowed under the frost line. And then as it thaws, it too pushes its way out and heads to the nearest vernal pool. So its tolerance for near freezing temperatures make it one of the first to awaken in the spring, and also that toughness makes it ideally suited towards the northern mountains. So I think that's a little bit like our humans who choose to live here for good parts of the year, that toughness and that willingness to withstand those cold temperatures. So as you're out there this year and you hear the air in the mid-spring full of sounds, make sure you take a minute to listen to the spellbinding chorus of the frogs. So with that introduction, we're going to head right into the meeting today. We're going to do some introductions for those of us who are in the room. I'm going to talk about our very cool Partner Accomplishments Dashboard and thank all of you for presenting to that data. We'll hear a little bit from the APIPP staff, and then we're going to turn it over from there to Nancy Budd from the Adirondack Garden Club, and we're going to learn about some great work that they're doing with the Adirondack Botanical Society and Plain Area Trails right here in this region. And then, as I said, we'll go to the partner roundtable, and hopefully many of you can stay for the field trip today. So I'm going to just start with a couple of quick hellos. And again, apologies to the folks on Zoom, but hopefully you can introduce yourself on Zoom. We're going to go around the room, and I'm going to actually just ask Ari to introduce himself so that we get that person, then we'll come back to the room. Yeah.
Hey, everybody. Ari Giller-Honeywell. I am the new invasive species, terrestrial invasive species manager with APIPP.
So I know Ari has not had a chance to meet a lot of you yet. So Rich, you want to just start and just say who you are and where you're from?
My name is Rich Ross. I'm with the Adirondack Garden Club Black River chapter out of Watertown, New York. We cover Lewis, Jefferson County, and Northern Oneida. We're close to one of the St. Lawrence Crew prison group. But I was coming to do an article for our newsletter on invasive species, and I'm just a sponge today to learn as much as I can.
I'm sure we can fill up many newsletter spaces for you. And 3-hour drive, so thanks for coming, Rich.
Dominic Peters, volunteer, Protector Corps, and Force Pest Hunters. Over to you.
Basically the same thing. Bill Wickering, I'm also, fire force specialist. I do a lot of work with Brian for the lake tracker on Upper Chattagay Lake.
And I think
Whatever else I can fit in.
Tom and Bill together, you guys have over 500. Probably, yeah, pretty amazing how they're looking for it. So, yeah, go guys.
Yeah.
Brian, go ahead, you got it.
Yeah, tough act to follow these rock stars here. I'm just a paid employee that does probably maybe a tenth of what these guys do sometimes, but I'm the Aquatic Invasive Species Coordinator, so I get to work with all of our streams and lakes and aquatic resources across the prism.
Zach Simic, Conservation GIS Analyst with APIPP. I am our data and map nerd.
Hi, I'm Mark Kubiak with the New York State DOT. I'm the new Adirondack and Catskill Park Coordinator, and I head up the Landscape Ecology Unit at DOT, and I'm looking forward to working with everyone.
Welcome, I'm glad to have you on board.
I'm Alex von Bieberstein. I'm also with New York State DOT and I work in Mark's group.
All right, we'll jump right to that. Oh, yeah, we'll catch you back in the doors.
Hi, I'm Matthew, the books in Lake George. Very grateful for the work you all are doing. Thankful that you're available to us.
I'm Susan Clark. I'm just a property owner on People Lake. And we've got a big push to try to control our species, and I'm sort of heading that up. So I'm here to learn and meet people and keep going with our project.
Great, up to Nancy.
So I'm Nancy Budd, and I'm from the Adirondack Garden Club.
And you'll hear from her later. Okay?
And I'm Laura Sells Doyle, also from the Adirondack Garden Club.
I'm Carrie Persian from Ausable River Association over in Wilmington.
I'm Aaron Zeman, Project Analyst for Forest Resources at the New York State Adirondack Park Agency.
I'm Steve Young, representing Adirondack Botanical Society today and also helping Essex Quarry.
Laura Laytonen, I'm a botanist. I've also been working with the Adirondack Garden.
Melinda Buff from the Adirondack Garden Club in the Cass Volunteer.
Guy Middleton, lake manager for the Upper Saranac Foundation.
Katie Whitkovitz, with the Hamilton County Soil Water Conservation District. We do a lot of outreach and some terrestrial.
I'm Chris Herron, Executive Director for Champlain Bay Trails. I'd say I started my environmental career in graduate school in Indiana, studying milfoil back in the 1980s. And started working with the Nature Conservancy in Indiana and dealing with invasive species throughout the state. So I'm interested in the state. Here we've got Champlain Bay Trails project at Essex Quarry that the Garden Club is essentially a project.
And it's fun to be in the CATS territories today.
I'd say it would be better at Terrestrial.
Perfect.
So anybody wants to see where they can.
Hi everyone, I'm Erin Bunny Ballrath. I'm with New York State DEC as the Lake Champlain Coordinator, and I work out of our Region 5 office in Raybrook. Hi everyone, Lauren Janess. I work for the Lake Champlain Basin Program out of Grand Isle, Vermont. I'm involved with our summer boat launch steward program, with our Streamwise assessment program, as well as a lot of grant management. Nice to meet you all.
Everett McNeil, East Shore Screw Lake Association, and focus on aquatic and terrestrial invasive.
Great. Again, thank you all for coming today. Ari, did you want to say another couple of words?
I'm gonna save it for when I.
We'll save it for your time. Okay, that sounds great. All right. So again, thanks to everybody on the phone. We got what? Dozen or more, 15 folks on the phone. So hello, everyone. We'll try and keep you going on the chat too. So I'm going to talk about the partner data dashboard for just a minute, because it is an awesome project, thanks to all of you. So several of you in the room were part of the strategic planning process that we had that led to our 2023 strategic plan. And there were a lot of surveys that we did also as well. And most strategic plans, as many of you know, just sit on a shelf. So, and what we fondly call shelfware. Although nowadays, it's not even on a shelf, it just sits in somebody's electronic file and nobody ever dusts it off till 5 years later. So we made a commitment when we did it that we didn't want our strategic plan to just live in an electronic file somewhere only to be dusted off. And it's not really just the strategic plan for the Adirondack Park Invasive Plant Program staff. It's really for all of the organizations that make up this amazing partnership for regional invasive species management. So it's pretty simple, 4 goals: protect land, protect water, engage communities, and engage in research. So the Adirondack Park Invasive Plant Program, APIPP, puts out an annual report every year, and we go through everything that we've ever accomplished. But there had been nothing prior to that rolled up. What is it that all of our partners are doing? How do we show the collective impact of what people are doing all across the park? And so last year we did our inaugural test of our partner data dashboard. And last year, we had 17 organizations report. Those of you who thank you for doing into the pilot. And this year we had 34 of our partner organizations to submit their data. And I'll tell you a little bit about how cool that is. It's also on our website, and I've got some copies to pass around if anyone wants to look at it. But this is the only place that we know of where all of the accomplishments of our partners roll up together. And the metrics are pretty simple for now, but if we get great take up of this over the years, I think we can add more to it. So what we know is that 16 of our partners were actively monitoring terrestrial invasive species, and of those 13 were managing them. And then we asked folks, which ones are you managing and monitoring? Purple loosestrife just turns out to be the winner of last year. So when it comes to aquatics, we have a few more of our partners monitoring for aquatics. There's 24 organizations doing that. And this was an amazing number to me. We have not, again, seen a roll-up of how many organizations in the Adirondacks are actively managing aquatic invasive species or their lake associations. We don't have the actual numbers attached to that at this point. Brian, I think you've talked some about like a ballpark number.
Oh, yeah, we're around $1 million a year.
Right, between all the lake associations that are putting money into managing. So this is really incredible. And there are 10 partners involved in operating boat stewards programs. Some of those are in collaboration with Adirondack Watershed Institute, and this also includes AWI's numbers. But some of the organizations here are putting in volunteer staffing in the hours when the boat launches aren't staffed. So a total of 200,000 visitor contacts in the Adirondacks last year. I'm astounded. And this little number about 2,000 and 2,100 interventions is like, okay, yeah, whatever. Well, that's over 2,000 parts of invasive aquatic plants or aquatic invertebrates, aquatic species that are caught before or after they leave, before they go into a water body, after they leave a water body, before they go somewhere else. Imagine if even a fraction of those had managed to get into new water bodies. It is just astounding what happens here. And it was so fun to see the data rolled up. Just in comparison, last year we had 6 partners reporting and really about half of the interceptions. So this was great this year to have a lot more organizations reporting into the dashboard. We had 21 organizations do outreach events, and we had over 3,000 attendees at those. This does not include the APIPP events, and we had close to 2,000 at ours as well. So I love those numbers. And then I think about 200,000 interactions with the boat stewards. It's incredible what the stewards program does. And we have 10 partners engaged in research programs. So I thank all of you for that. It is great to have a chance to share that with you, but when I sit down, I'll pass around this year's dashboard. Again, it's on our website as well. We encourage all of you to take that back to your organizations, right? It's not just about us. We want you to be able to say, hey, we're one of the 13 groups who's managing our aquatic invasive species here in the Adirondacks, or we're only one of 13 who's actually taking care of terrestrial land. So this is important for all of you. We ask any of you who are not a formal partner of APIPP, there's a list on our website. please become a partner. All we ask is that you show up at one of these, or you contribute data to the dashboard, you participate in a working group, really anybody who's working on invasive species, and you get a chance to connect with great people, access to lots of expertise and recognition through the dashboard, because everybody who has participated and contributed to the dashboard, all of your organizations are listed. So I'm going to just quickly go into what is it that APIPP is doing this summer. And very fortunate that we have 4 of our 5 full-time staff here today. Sean is not with us because he's on paternity. So Cormac Xavier Kittle was born in mid-March, and Zach will be back next month. I mean, Sean will be back next. Zach is not a father yet, just wanted to congratulate him at this point. You can congratulate him because he got married last year, but when you see Sean, you can congratulate him. So we also have 3 outstanding seasonal employees that Becca Tamagna is coming back for our 3rd summer, and she does all the management of the DEC operated campgrounds and a lot more. Dana's our aquatic specialist, and Reed Middledorf was with our pro crew at Invasive Plant Control last summer, and he's coming back to do terrestrial work and forest pest work. So I can't be more delighted that we have a lot of great people coming back this summer. So since Sean is not here, I'm going to flip our goals. As I said, 4 goals: land, water, community engagement, research. Pretty easy to remember. But since Sean's not here, I'm going to talk just a second about the community engagement piece. We have, as many of you know, our Protect Your Forests and Waters posters, as well as the brochures, and then the new terrestrial field guide. We've got a lot of those with us today, so if you need a stack of 100 to go stick in a kiosk somewhere or to distribute at your meeting, please go right ahead and take what you need today. I don't have all the posters here, but you can order them for free on our website. You just go to order outreach materials on one of the drop-down boxes. We usually have got boxes of them at the office. They're really designed to get out into the hands of people who are out there doing invasive species prevention messaging. So please take advantage of those. I also put in the back, we have just updated our native plant supplier list in honor of spring. So if you're getting out and doing some gardening and you want to look at where can I order some native plants in addition to the Adirondack Action or ADK Action Pollinator Project, here's a list of gardens and garden supply centers that have native plants. We have really fun and exciting events coming up. So on May 29th, we're going to do a webinar, and that means you don't have to drive 3 hours to get here, but we're going to look at 4 species that are not yet in the Adirondacks that may be coming. So that's the May 29th webinar. And please mark your calendar and hone up on your trivia skills for June 5th. We're going to celebrate New York's Invasive Species Awareness Week with an event with Racket River Brewing. They are producing an IPA, which is for invasive plant ale, not for India Pale Ale. It does not have milk oil, though, in the actual mash mix as I understand. But we're going to have that over at Racket River. We're taking over their Wednesday trivia night. It's going to be a blast. I hope many of you are a chance to come.
So I get a 6-pack and I do a Zoom.
Yeah. Right. Those of you not on Zoom. So the question was whether we could get 6-packs to those people who might join by Zoom. And the answer is we have not worked out the drone technology for delivering the 6-packs yet, but hope to see you there. And really, really hot off the press. October 10th, we're going to be doing our biennial Adirondack Invasive Species Summit. So we're going to be at North Creek. We're working on the logistics with Tannery Pond. Really amazing lineup. Beth Bradley from the Regional Invasive Species and Climate Change Network is going to come talk about invasive species and climate adaptation. We're going to have someone come talk about New York's climate assessment, where it touches down here in the Adirondacks. And then we're going to blow your minds with a lot of cool things about genetics, but you don't have to know genetics. We're going to look into all the different types of eDNA that are out there. If you want to get into that, how do you pick what's the difference between a qPCR and a metabarcode? We're going to distill all that so we can all understand it. We have someone who's going to talk about manipulating the RNA of Phragmites so that it, so to kill the plant. So a natural biocontrol, if you will, with a little gene manipulation for Phragmites, and then wrap it up with one of the researchers at the University of Connecticut who is trying to work on hemlock resistance and is mapping the hemlock genome and has found out very cool things about what our hemlock trees are doing at various times of the year. So please come on October 9th. So with that, goal one, I'm going to turn it over to Ari to talk about coming up on our terrestrial work. And again, lunch, if you want lunch, stick your lunch tags in the back.
So this is week 4 for me as terrestrial invasive species management. So a lot of these things I'm excited to dive into more as the field season starts. To give you a decent overview of what we're up to on the terrestrial invasive side. New species that are emerging in the region, forest pests, season plants, all that, and the training continues. First, little info about myself. Just relocated to northern Vermont. Really excited to join people. All such an engaged conservation community, and I feel like I've been looking at it the outside. Past 5 years, I was in the private sector as a residential arborist. Plant out there. Certainly no shortage of invasive species, but more on the yard scale versus the greater landscape. From DC, an environmental engineering degree from James Madison, Shenandoah Valley, and worked with the Park Service on different exotic plant management teams. DC, out of the east, and that really encompassed the Sonora and Mojave Desert. Some endangered species surveys in Utah and in other areas. But most recently, the lower Hudson Prison went with Trillium and Green Cross is where I think most of my residential for verticulture together with Hudson. So my partner and I relocated the team, and we're super excited. So in terms of some of what we're going to be tackling this year that's not widespread in the Adirondacks, put it that way. Line barrier, find right now is Single Island in Lake George. For treatments that are not approved, we're going to go out. Mile-omitted is another species where there's not a lot of established population. That's going to require coordinating with private landowners. And we really want to keep that from spreading to the region. Swallowwort, there's a little bit more, but I'm excited to hit the ground running within the next several weeks in terms of early technology we're going to get out and start this. This is a great slide in terms of folks that aren't super honed in with the different invasives, things to look out for. Giant hogweed is another one worth mentioning. There's not a lot in the Adirondacks, but it's a very coordinated effort across the state. So I'm working with Nick, Naya Krause. There's, I think, 3 or 4 sites total in the Adirondacks. Thankfully, many are just down in the monitoring stage, but it's another one that's a successful detection route in this one. Grouses, we also want to keep our eye out for coming from point south valley. I certainly noticed still grass on a lot of trails, trailheads, and that it's being transported with people. I definitely think it's not real education to make sure that people aren't spreading it or recreating. Carpet grass too, not widespread, but because of how they spread in total colonies, treating them early versus when they're super established, it's going to be a good strategy. Tree of Heaven is another one. You can see on the map, it's not in the region at large, but definitely going to focus on getting last landowner permissions around the southern end of Lake George. Single tree can have over 3,000 seeds a year. So even though it's not widespread, we can address this as soon as we can. tie into Tree of Heaven, it's the preferred host plant for spotted lanternfly. As far as I know, we don't have any spotted lanternfly in the Adirondacks, but I really appreciate all the partners that are going to be helping traps this year. This is one where instead of deep in backcountry areas, I think right along highway corridors and people transporting his vehicles, it's a concern. So I'm excited to coordinate this project. Here are the folks that Sarah mentioned. They're going to be doing the bulk of the work for the terrestrial program in terms of survey and treatment this year. I'm really pleased that Rebecca Tenango is coming back for year 3. I'm sure she knows way more details about these DEC campgrounds and survey sites than I do at this point. So I think we have a lot to learn from each other. Reed actually was on the pro crew last year and is now an invasive species assistant. So I'm really excited to be working with him on a lot of the projects I'm talking about and we'll get into with different biocontrols and trapping. In our pro crew, their main focus is knotweed and Phragmites, but they're working on a variety of different projects. I believe we have them for about 3 months this year, starting in July. So thank you to Forest Pest Hunters. I haven't met Bill yet. It's great to meet Dom. You guys did amazing work this winter. And you can see some of that. We use IMAP invasives. If anyone hasn't heard of IMAP invasives, it's a great app, great mapping program for both agencies and just anyone to use. It really helps paint a picture of what's going on, who's finding what where, and compiling it all in one central place. At some point, I'd love to go deeper into the mortality studies. Those still are ongoing, but certainly as winters warm, the concern is if we don't get these cold temperatures that kill off the majority of adelgid, is that going to facilitate the spread? In terms of the spread, I'm excited to hear from Zach in terms of modeling and priority areas. With this data, how can we integrate that into management plans moving forward? In terms of getting the data, Forest Pest Hunters are an amazing resource. We have an adopted trail program, and then early detection, like someone thought maybe they found it in the Faro Mountain area. So one or both of you went out and immediately checked and did not find it, but that's just that immediate response is so appreciated. It helps our program so much. So we'll definitely, I have a link here in terms of more details on the program. Hemlock adelgid surveys ended a couple weeks ago. The concern there is the crawlers might be starting to hatch. So the information is great, but the potential spread of the adelgid, that's why we cut off the surveys in the winter. Beech leaf disease is going to be the focus of summer Forest Pest Hunter survey. This is definitely a concern just in terms of how quickly it's spreading. And beech trees, pretty darn important in terms of the masts for wildlife and how much of the forest they encompass. It seems like that disturbance certainly could be a pathway for new invasives. So both understanding what's causing beech leaf disease, the nematodes and the buds is definitely the current theory, but also how can we treat and reduce the spread? So certainly more to come on this. Emerald ash borer, we're also happy to be in our second season of biocontrol releases with APHIS, as well as a handful of study plots where there's no known emerald ash borer to track the northern progression. And lastly, I'm hosting a spring partner training day. I emailed some of you about it, but if any of you or your staff wants to join, feel free. I'm going to be going over invasive species ID, different treatment techniques. And Sean's also going to be there leading a photography workshop. So definitely get in touch with me if you haven't heard about it already and you're interested in joining or having your staff join. Really excited to be working with all of you. I know that was a really quick rundown, but be in touch anytime and you'll be hearing a lot more from me in the future.
All right, we're going to switch gears a little bit and we're out of order with our strategic plan, but I'm going to give a really quick overview of some of our research and innovation work that's underway. And just one quick housekeeping note, if you took a sandwich form, we have a completed stack of forms started on the back left corner of that table back there. So you can drop your sandwich slip there. All right, so what are we working on for research and innovation this field season? We'll be continuing some of our sort of long-running existing research and monitoring projects, including MAMA, or monitoring and managing ash, AVID, assessing vegetation impacts from deer, and we'll be starting a new hemlock health plot monitoring protocol this year. So just a quick overview if you're not familiar with these programs. The MAMA, or monitoring and managing ash program, is a system that's designed to identify lingering ash that could be potentially resistant to emerald ash borer. We have a couple of different plots set up in different parts of the Adirondacks, waiting for EAB to reach those areas and monitor the decline of trees in those plots, hoping that we'll find lingering trees. Assessing vegetation impacts from deer, or AVID, is an established monitoring protocol to identify areas with overabundant deer that are having negative browse impacts on our native vegetation. Why are we thinking about deer for invasive species management? The answer to that is where we have a lot of deer, it can exacerbate the presence of terrestrial invasive plants because the deer are eating all the nice native vegetation and only leaving behind all the invasive stuff. So if you live or work in the Champlain Valley, you probably have seen some evidence of deer exacerbating terrestrial invasive plants. And new this summer, we'll be establishing some hemlock health monitoring plots. This is a protocol that Tamara has been heavily involved in developing with a coalition of partners from the Nature Conservancy and other organizations. And the intent of these hemlock health monitoring plots is very similar to to MAMA, where we'll be monitoring the health of hemlock trees over time. And when hemlock woolly adelgid shows up at some of these plots, we'll monitor the decline of trees in the hopes that eventually we'll be able to search for lingering trees or resistant hemlocks within those plots. This is going to be a very long-term monitoring project here in our region. And then we're continuing two existing ongoing research projects in collaboration with the New York State Hemlock Initiative that is evaluating the use of eDNA as an early detection monitoring tool for hemlock woolly adelgid. This will be our third year evaluating that technique. We've had mixed results at best so far, but we hope that maybe we can refine that protocol to something that is a little bit more practical in the field. We're also involved in a really interesting project that is evaluating the movement of pesticide from treated hemlock trees to nearby adjacent untreated trees. So trying to have a better understanding of what are the potential negative impacts that we could have with some of our chemical treatments. Acknowledging that that is an important tool in the toolbox and one that we need and want to retain, but want to have the full understanding of how it could potentially impact our environment. So we'll be hopefully wrapping up that project by the end of this summer and share some results with everyone, maybe at our fall partner meeting. And then I wanted to take just a few minutes to share some information on a really exciting to me, ongoing project that Tamara and I have been involved with a coalition of partners from the Lake George watershed and beyond. We're calling the Lake George Hemlock Coalition, in which we're working to establish what we are calling priority hemlock areas, or PHAs. So what are they and what is their intent? As you can see in the map on the right-hand side of the slide here, with the cool animation.
Very
I
nice.
did say math nerd when I gave my introduction. So this is the distribution of hemlock woolly adelgid in the Lake George watershed. Currently the only part of the Adirondack Prism where we know HWA exists. But as you can see within the watershed over the last really 3 years since the first established infestation was detected on the eastern shore, the distribution of the insect has really expanded. So this is beyond the point of eradication. This is beyond the point of containment. So now we're really in a place where we need to focus on suppression and resource protection. So with limited resources available to actually manage HWA on the ground, where are we going to focus those resources to protect trees that are most important? And that's the whole intent of establishing these PHAs. So in summary, PHAs are high value stands that are important as identified by the coalition. And the long-term intent is that we will be monitoring these stands, and should HWA show up, we will be doing some type of chemical or biological control action to help protect these important trees. So we went through an extensive planning and modeling process to identify some of these stands that engaged a group of regional stakeholders to brainstorm criteria for what defines a really important stand in the watershed. And this process is data-driven. It is something that is both replicable and scalable. So eventually we may be able to take this beyond just the Lake George watershed should ACAA show up elsewhere. So we've included about 16 different data layers or criteria that we're using to identify our priority hemlock stands. I don't expect that you can make out any of the text in these maps, but we're thinking about things like slope and soil erodibility. Should trees decline, we want to prevent erosion. We're thinking about proximity to hemlock-dependent species or proximity to rare species. We're thinking about the conservation or protection status of these lands. A whole suite of variables went into this model that were developed by the coalition and weighted for the relative importance. And what it has resulted in is this comprehensive score map that basically casts out across the watershed how important is each individual pixel of land area that contains, in this case, conifer. It's not truly hemlock, but conifer forest. Where in this map, the darker shade of green, the darker the green, the more important that stand is. And the darker the shade of brown, well, maybe that's what we're going to write off. That's a little bit less important as far as our model goes. So this is really where we're at now with the coalition. We have our comprehensive score map pretty settled. And what we're working on doing is actually citing or locating these priority hemlock areas. Where will they actually be on the ground based on some parameters and criteria that our coalition has developed? So thinking about how big can they be? How small can they be? How far apart can they be? How close together can they be? So we're running through a couple of scenarios now. These are all just draft versions where these polygons represent potential priority hemlock areas. But over the coming month, the coalition will finalize these results. And we hope that by field season 2024, we will actually have some boots on the ground going out to visit some of these really high priority stands. And moving forward into the future, I think there will be a great opportunity to engage some of our forest pest hunters and volunteers to visit some of these. I know how much Bill loves walking along the Tongue Peninsula, so I'm sure he already has his eyes on this. And that's all we have for research and innovation. I'll pass it to Brian.
Well, if you're like me, we got all the boring stuff out of the way. Now we can really focus on high quality, important aquatic stuff. I see a lot of our aquatic friends here. So will admit my bias. I love our lakes and our streams up here. Our forests are great things that provide great benefits to our lakes and streams. So we do need to protect them as well. But I'm glad to be talking to you here today about all the aquatic work that we do together as a team. And yeah, really impressive to see those the dashboard numbers and look at everything that collectively do. I always say that, it's a one place, AWI puts out their report, or their boat stewards, like George Park Commission puts out theirs, the Lake Champlain Basin puts out their separate one, maybe some towns are doing. So one place to really be able to roll that in, and the numbers are incredibly important. And that's because we have one of the least invaded aquatic systems in the country. And so we are trying to focus a lot on prevention and trying to protect that. We're going to do kind of like a two-part talk here. I'm going to kind of give you our 2024 AIS programs, and then I'm going to dive into eDNA, talk about what we did in our 2023 project, and talk a little bit about where we're going in 2024 with it. So I'm going to start off talking about our Lake Protectors program. We have several lake protectors here in the room. This is our program that has been running since 2002, where we trained our partner organizations and members of the public how to identify and report 16 species, 8 animals and 8 plants. And they adopt a local water body, or in Dom's case, many water bodies, and they go out and they do a survey at least once between July and September, and then they report back to us what they saw. Last year was a record year. We had 184 surveys, so that is a very high bar for us to try to stay up with again, but hopefully with all your help, we'll be able to do that. We will have 3 trainings in June. We will do an online virtual training at the end of June. And then we do 2 in-person ones. July, it's gonna be in the Caroga area. So you'll be able to, you know, we bring live plants, we bring preserved specimens, we go out on the water, so, you know, really hands-on. So we're doing one in July in the Caroga area, and then August in Newcomb at the SUNY ESF Adirondack Information Center. We'll be doing that, so be on the lookout for upcoming dates for those. We are also continuing our lake management tracker program. This is a program that Aaron started in 2018 along with Lee Walrath. And it's just a really, really cool program. We're now going into our 7th summer of it. And it is for communities and lake associations that are actively trying to manage invasive species. Most of these are Eurasian water milfoil. We do have some that are doing variable leaf milfoil. And it's really designed to help them understand how effective their management is and if they're reaching their goals for it. And what it is that it's a modified point intercept monitoring location where they go out to the same place and they ask the same series of questions. And by doing this year after year in the same location, it kind of gives us this overall census. So kind of like a dashboard of like what's going on with milfoil in there. APIPP provides all the technology and the training. The local communities provide the people power to go out there and do this. Bill has been an incredible volunteer for years on Upper Chattagate. And yeah, so we will continue this. And like I said, because we're doing the same thing year after year, our first lake that Aaron started with was at Loon Lake. And so you can see that, you know, over 6 years, their management efforts have held milfoil to only increasing to 5% of the locations. There's been some really interesting data we've collected for some of our other lakes, by like Paradox Lake, where we've seen really large increases in it, and it's kind of made them use an adaptive management framework where they're using this data and saying, well, we kind of got to change some of our control management efforts because it doesn't seem like we're getting to where we want to go. So we will be continuing on with our existing lakes: Chattagate Lake, Friends Lake, Lincoln Pond, Loon Lake, Moody Pond, Paradox Lake, Racket Lake. They've been in the program for years now. And we have 2 lakes that have signed up and said they're going to start new. So Eagle Lake, right here, Susan's excited to get started this summer. And Peck's Lake, and there's other lake associations or communities, you know, that are doing milfoil management. This is a really good tool to empower them to get better data to help understand if they're reaching their goals. So if you know anybody else who's interested, you can have them contact me. Another great project that we did last year in 2023 and that we are able to continue in 2024 thanks to the Lake Champlain Basin Program and their funding that they get through the EPA is this, we call this Lake Champlain Boat Launch project, where we worked at 5 boat launches along Lake Champlain that were 5 of the highest boat launches for AIS leaving the boat launch. Unfortunately, we'll never be able to control or reduce all the invasive species in Lake Champlain. It is our most invasive water body in our prism. But the idea was, could we work at that very local level, right at the boat launch, to be able to reduce the amount of invasive plants, Eurasian water milfoil, curly leaf pondweed, to be able to reduce the amount of invasive species that are leaving that boat launch and potentially going to other lakes and as a vector of spread. We are building on top of the great database that our boat stewards collect. Every time you go to a boat launch and there's a boat steward, they're asking you these questions. They're putting that into a database that we call the WISP, and we can actually track this. So you can see these are the 5 boat launches, and this is the percent of Eurasian water milfoil on leaving. So this one year at Ticonderoga, almost 25% of the boats leaving there had Eurasian water milfoil on them, which is not a good sign. But one thing you can see is that the data is really variable. And there's a lot of interesting things about the variability. There's variability from year to year because of seasonality, how the plants are growing, use, how the use of these boat launches, the popularity of it changes, and then also some the difference between boat stewards actually does make a difference too. So interesting, in our first year, we kind of saw that We learned a lot. We got a really great amount of interesting data. As you could imagine, 1 year of doing diver assisted suction harvesting at a boat launch is not going to solve our problem. So we did 5 boat launches last summer. We learned a lot about those individual boat launches, and some places it worked well, and some places it didn't work well. So this coming summer, we're only going to continue the management at 2 of the locations. We'll continue monitoring and tracking at all locations, and hopefully at the end of next year, I'll be able to give you another full project update on that. A new project that we are starting this year is about milfoil management and water quality. Frequently, I get questions about, hey, what's the impacts of managing milfoil, both hand harvesting, diver assisted suction harvesting, and now upcoming chemical management. How does that impact the water quality? What does that do to the pH? What does that do to the nutrients levels in there? And quite honestly, there's not a lot of data out there that we can look at there. Now, logistically, when we're talking about this with other scientists and other different things, our working hypothesis is that if done correctly, that our management should not be impacting the water quality. But it'd be good to have some data to back that up. So we started to look around at this, and we found that out of a lot of the lakes that are doing management, have been part of the Adirondack Lakes Assessment Program called APIPP. It's a project, a program between Protect the Adirondacks and Paul Smith's Adirondack Watershed Institute, where people have been collecting water quality data for decades in some places. And so we could really piggyback on that because we had this long historical data that these lakes have been using to monitor their water quality. And so we are working with 5 lake associations that have been managing invasive milfoils for a while. And we're also going to do monitoring on 2 water bodies, Middle Saranac and Follinsby, which are not doing management on those, not doing management of milfoil. And then here's just showing a graph showing, there's all sorts of different parameters they're tracking, and so we'll be able to track how these things fall in their trend line. And so the idea is that traditionally in the APIPP program, they've been going out and they've been monitoring at the deep water, the open water hole, generally kind of somewhere in the middle. This is Chavey Lake right here. All these little green polygons are places where we've mapped beds of Eurasian water milfoil. So they've been going out and they've been monitoring this location for years. What we want to do is we want to add in 2 additional monitoring locations. One would be what we call a milfoil treatment. So they have been doing diver assisted suction harvesting at this bed for several years. So we're going to add a water quality monitoring location there. Then we also want to add another water quality monitoring place at a shallow water area that has milfoil, but that is not being managed, so that we can then be able to compare these 2 sites to each other, and then also to the traditional open water monitoring area. So we'll be going out from May through September, collecting water quality samples and sending them off to Paul Smith's to be analyzed. We will also be doing our early detection surveys. Since for 9 years, I think this will be the 10th year, we have been contracting with professionals to go out and do monitoring for early detection of species on lakes. We have covered now over 170 different lakes where we've had our professional crews go out and monitor in that. And a big focus of that was collecting data and high quality mapping to go into a modeling project that we wrapped up last year for our vulnerability modeling. So you can go on our website and you can look at areas that we were able to use a data algorithm to predict which areas are most likely to be invaded by invasive plants. So that was a really successful project that we wrapped up. And so we are sunsetting our professional contracts that we're doing of that. But our APIPP staff, we will continue to go out and do early detection monitoring. So myself, our seasonal assistant, Dave Homeland. So if there are places that you need help, people looking at you hear about things, please let us know. But we will have the team from Adirondack Research. They will be going out and doing 10 lakes this summer, and I'll talk a little bit more about it. Yeah. So any questions about this or overall 2024, any of our subjects before I'm going to jump into another segment talking about our eDNA monitoring programs?
Water quality monitoring. What program?
ALAP has a set suite, so we wanted to go with a set program so that people can continue in with that. It is they have a standard suite of things like nitrates, phosphates, pH, the color. There's around a dozen different ones that they're doing, and these are integrated 2-meter samples. So we're putting a tube in the upper 2 meters. And then you're taking that and you're filling that. We also do a chlorophyll A sample, and we also do a Secchi disc reading for it.
Right, would it make sense to combine ALAP and CSLAP into one program? Is that any possibility?
So logistically, no, it can't work. So ALAP is the Adirondack Lake Assessment Program. It's a focus on the Adirondacks. It has a couple parameters that are a little bit specific to us. Like they measure chloride. So when you've heard about like these road salt, like why we are concerned, all that road salt talk that came out from ALAP people monitoring lakes and signaling the alarm that, hey, our lakes are getting a lot more chloride in. So it's a little bit tailored, and I don't think the people in Western New York want to take part in the Adirondack Lake Assessment Program. So that's how CSLAP kind of goes around. They measure a lot of the similar things, but they're not exactly the same. So this CSLAP, Citizen Science Lake Assessment Program, that's our statewide program that's run through the NYSFOLA group and DEC. Yeah.
I'm curious about the water quality monitoring too. Are there any lakes that are other treatments aside for the dash harvesting that are on that list that you're thinking of?
Yeah, so hopefully, let's see if we can go back here. Kuroga is put in for an application to do chemical management that is going in front of the APA next month. So they potentially will be doing a chemical management. They've been doing dash in the past. Chattagay Lake is also put in a permit application to do chemical management, so they also might do chemical management this summer. They've traditionally been doing hand harvesting. Chazy is doing diver assisted suction harvesting, and we'll continue with that. Paradox has a permit to do chemical management, and they are planning on doing chemical management, so that one we will be monitoring chemical on. And Racket does hand harvesting variable leaf milfoil. All these, Racket is variable. All these other ones are Eurasian milfoil.
And then it's the Follinsby, is that Follinsby Clear Pond or the Follinsby Preserve?
This is New Follinsby Preserve from the Nature Conservancy. That has no milfoil on it, thankfully.
Call it the Adirondack Basis Program. Are you looking at a range outside that blue line that like pretty deep apart that affects the water monitoring? It's like a 20-mile.
Yeah, so our prism, the Adirondack prism, is the Adirondack blue line, and then Franklin and Clinton counties going all the way north to Canada. So then they broke the state into 8 different regions. So to our south is the Capital Region, to our west is the SLELO, St. Lawrence, Eastern Lake Ontario region. And yeah, we work very closely with them to look at things that are coming up on the horizon. And we have some of our projects, like that vulnerability mapping project. We actually map out all the lakes 10 miles outside of our prism. So yes, we do try to work and pay attention all along that area.
Is that map of where emerging species might be on the website? So you could, if you were gonna go to a place and canoe, you could look at it.
Yep. Yeah, we can pull that up at one of the breaks or something, if somebody wants to look at it, or you just Google APIPP vulnerability map, there's the first thing that comes up. Any other questions for the other topics?
I wanted to offer one other piece on the Lake George Hemlock Coalition work that is getting going. That a part of that, thanks again to the Lake Champlain Basin Program, the Regional Planning Board for Lake Champlain, Lake George will be actually taking on and doing more mapping and community engagement work this fall. But as part of that, they also will be pulling together a bunch of resources for private landowners. And we hope to also get some of that information on our APIPP website as early as this summer, because we know there are a lot of private landowners who we don't want people going out and saying, oh my God, hemlock adelgid's coming. I'm going to cut all my hemlock. So before the adelgid gets to it. So we will be working on pulling together some of the latest science on how to manage hemlock. And one of the partners in that coalition, the Warren County Soil and Water Conservation District, working with the Washington County Soil and Water Conservation District, is planning a field day for landowners on September 21st at Chinchacook. They'll be bringing in some of the Forest Service silviculturalists and some others talking all about how to manage hemlock. So there's a lot happening on that front as well.
Tamara, is the mapping inclusive of private lands as well, or is the mapping of priority hemlock just on state lands?
Yeah, so the mapping is cast across the entire watershed. One of the variables that's part of the model is ownership, and it is weighted more heavily towards public land as being more important. Just because of the long-term protection status of like forest preserve lands, we wouldn't want to invest in treating trees and then have them be cut down on private land. So that's the reason for the weighting. But that being said, these blobs could theoretically show up on private lands. Would we necessarily engage with those as like our first go-to? Probably not, but they would remain on their list. Yeah.
Right. And I think what's important about that is that ultimately these will be places that are prioritized for monitoring and then eventually for treatment. And when we talk about treatment, that means either release of biocontrols or chemical treatment. It's really expensive. It takes a lot of human time. We're able to collectively with DEC, APIPP, some private applicators in the region, only a couple hundred acres a year are ever able to be treated, and there's a finite supply of biocontrols. And so DEC is the lead partner, especially in doing the treatments. And so from the state's perspective, if they're going to be investing state taxpayer dollars, that needs to go to either state lands or lands that have some long-term protection, and not to then treat a bunch of hemlock trees that may end up then being harvested when the landowner needs to be able to monetize their stand. So, and again, this is just a really early foray. It has not been done in New York. It's certainly been done in other states like North Carolina, where they have a lot of hemlock adelgid mortality. So we're really delighted that DEC and again, a bunch of other partners are even engaging in this conversation. So this is like literally very early stages. There'll be a lot more information over the next couple of years.
Do we have an idea of how effective the treatment is? And you know, it's a long-term investment, right? If it's all around it.
Yep. You want to talk to effectiveness of treatment?
Yeah, I mean, so the treatment itself, the chemical application, there are typically two products that can be used, both of which are quite effective. One is fast-acting, short-lived. One is slow-acting, long-lived. So it is very effective at controlling adelgid, but it is slow to apply. And within 1 acre of land, you can only treat 256 diameter inches of tree in a single year. So when you think about like you have a mature stand with 20-inch trees, that's a very small amount of the forest that you can actually treat each year. That said, once you treat them, they're protected for anywhere from 5 to 7 years before retreatment is needed.
And private landowners can work with private applicators to have those treatments done on their properties. And the reason you have that diameter limit is not something like we came up with, right? It is actually part of the regulations that go with that pesticide. It is part of the labeling limitations on that particular pesticide. So, yeah, we go out, we measure every tree, and figure out how many are in an acre. Yep, that's why it takes a long time. I think we have time for one more. Oh, yeah, Lauren, go ahead. Maybe because you tied on your hand.
It looks like on the map, where the Hemophilia delicate is being found is also in those green high priority areas to manage for. So I was wondering, like, does that impact like the speed of this project over?
Yeah, so interestingly, in an earlier iteration of the model, we actually included the location of HWA as one of the criteria that prioritize stands. We have since removed that, so it's actually not influencing it, but as you observed, oftentimes the dark green is where there already is HWA.
Right.
So I think it might, in some areas, impact like how quickly we're going to engage in those sites. For some of these, they're going to just be monitoring locations for quite a few years, we hope, knock on wood, because there is an HWA there. But for some where the PHA area overlaps with where Hemophilia delicate currently is, those might be candidates for some type of management action, you know, even as early as this fall.
Sorry, Brian, we got into TIS here, but
Yeah.
real quick, what does it cost to treat like one of those plots, the little red dots? Thousands of dollars?
So as far as the cost of treatment, well, first off, it depends on if you're doing it yourself or if you're hiring a contractor. And a little bit depends on like the topography and how difficult it is to work with the site. There have been estimates that range anywhere from like less than $2 to over $8 per diameter inch for treatment, depending on the site.
Per tree, I mean, what if you had a plot with like 50 trees, what would that cost?
I have spoken with some private applicators that rather than like an inch pricing, have shared that it would probably cost about $1,500 to $2,000 per day of treatment. So I'm a pretty fast applicator. I think under ideal circumstances, I might, like with good support, treat 7 acres a day. That would be quite a bit.
And that, yeah, so we can get more into that for anybody who wants to talk about that, but this is, you've got multiple people out on site, you've got one applicator, somebody else is measuring, taking the data down, making sure you haven't hit the limit, ferrying water back and forth. It is a team event out there when that happens. I did want to, I'm going to turn it over to Brian in just a second, but some of you saw a quick message I sent out yesterday on the APIPP listserv. We are absolutely delighted that the DEC has freed up funding for a 2-person hemlock crew to be hired by the New York State Hemlock Initiative. That's going to be working out of Warrensburg, out of the DEC office. It's going to be working in the Southern Adirondack region, not exclusive to Lake George. As far as we know, this is the first time that this kind of conservation team is kind of coming from DEC funding, but getting parked in an entity like Cornell and the Hemlock Initiative. It's similar to some of the aquatic positions that DEC is now putting into Cornell as well. So this is just great news. And if you know of any pesticide applicators who want a full-time job working out of Warrensburg, we hope that they get a great candidate for that position. We can talk about this all day, and some of us will as we're out on the field trip, and but we're going to turn it back over to Brian for the sort of showcase of what happened with, we're switching back to aquatic eDNA.
Yeah, and if you're interested in learning more about Lake Management Tracker, next Friday at the NYSFOLA annual conference in Lake George, I'll be giving a full talk just about the Lake Management Tracker program. So hopefully, Aaron, you're going to be there. Okay. All right, so we're going to go into talk about eDNA, environmental DNA monitoring, give a review of our 2023 project, what we did, how it was designed, what we did, the results of it in full detail, and then we'll talk a little bit about what we're going to do in 2024. So you probably have heard people saying eDNA a million different times. It's not EDNA from the, when you type in eDNA and you hit that in Google search, it gives you an Incredibles character first. It's actually environmental DNA. And so what that means is all organisms, plants, animals, insects, we are shedding off cells, skin cells, parts of the organisms, animals, their waste, their gametes, and all that contains genetic information in that. And so it's all here in the atmosphere. So we can collect that, and through some fancy lab methods, we can analyze it and detect the genetic presence of different organisms because we know their genetic code. This environmental DNA that gets out, it's impacted by things like microbes and time, the pH, temperature. So there's different amounts of it will degrade or be able to stick in different environments, but it's becoming a very standardized monitoring project. Carrie's been doing it for years with trout and salmon all throughout her watershed. So it's a really useful tool, especially for animals. We'll say it works better for animals than it does for plants. And so why do we do it? Well, it helps identify small populations. Research has shown it can traditionally find some of the organisms by eDNA monitoring rather than traditional monitoring like nets or visual samples or equipment-based things. You probably didn't even notice, like when you're looking at this picture, like these little plants right in here, a few plants, that's European frogbit. So sometimes it's kind of hard to find those little initial populations. It can be very cost effective when you're looking at all the time and effort we put out there, and it uses standardized techniques. So it can be replicated in time and also in different locations with the same methods. So in 2023, we started a project, and we had these two main questions. One, will we detect any new species to the region or to the individual lakes that we are monitoring? And the second question is, will results be able to identify aquatic invasive species already known to be present in the lakes? So how does it work? We went out to like near shore, popular high recreation use at boat launches, beaches, marinas, and we collected 250 milliliters of water. You're just literally taking your hand, putting it in the water and filling that bottle up. That's it. That's your random grab that you got, and whatever DNA is in that 250 milliliters, that's what you got. It then gets pushed, all that water, 250, gets pushed through a syringe into a filter that is right in there, and then that filter collects all the cells and organic debris and different things on it. And then you put it into a preservative, it gets frozen, and it goes into a lab. We work with the Cornell University eDNA and Genomics core facility. And we were working with them because they were doing a unique type of test that's called a multiplex assay. And each one of those little tiny samples got tested for 54 different species, around 25 animal species, and well, it was around 20, it was about even, it was about half this group. There were some marine species in there, so saltwater species that we're not going to detect in that, but it was a standard. Why we chose this is because, one, we're testing for multiple species within one sample, and two, it was developed as part of a statewide project. So we're using that same test in different places in New York, so we can compare apples to apples results on that. And so get into the kind of like the weeds a little bit if you kind of want to know a little bit how it works. We collect a water sample. In that water sample is genetic material. That genetic material gets sliced in the lab and broken out and freed. And into that, they put what we call bait targets. So we have these known segments of the DNA. So we put it into that same sample, but it has a molecular marker on it that we can tell the difference of it from the ones collected in there. And then it goes through a PCR machine, which is doing a lot of runs and amplifying it. So it's amplifying the DNA the bait DNA that we put in there, and then also the DNA from the environmental sample in there. This helps us it allows for some quantification of it. And it helps do some of the catalysts for these rare, smaller ones in there. So we then take that information from all those samples that have been amplified, and it goes into sequencing, and we're able to kind of identify these different markers And basically what they're kind of doing is they're having each species has a unique segment, this code. That, you know, your A's and T's and G's and C's, and so that's like your special little marker. And then it's looking through and saying like, okay, this little fragment right here, nope, this A, it's an A, not a G, so it's off by one marker. And this one up, we got a little bit more of it, you know, and then, oh, you know, here's one, you know, that got a full length of it. So we're trying to match this unique snippet of code. And there's specific markers for each species from a specific gene. And we'll talk a little bit about this when we get into the plants versus the animals. There's a little on the animals, the plants do not, the markers are not as specific. Okay, so how do we choose where to monitor? We use the New York Natural Heritage programs. They have a statewide analysis that they did where they ranked lakes being different levels of invasion risk. So we chose the 22 highest lakes that in our region that had public boat launches on them. And so this is a map just showing some of those locations. At 21 of those, we did one single location we did. Then we were very lucky to partner with Ebb and the Scroon Lake Associations. At Scroon Lake, we did 9 locations within the lake. So in total, we collected 89 samples, 21 samples, including one blank, field blank at Scroon Lake. Here's Scroon Lake, that's what you can see, it's just got a lot of them on there. And then 21 other lakes, there were 68 total samples. So Upper Chattagate at Bill's house, we collected some. Cranberry Lake, Lake George, Trout Lake, Indian Lake, Coroga Lake, Peck's Lake, around there, Great Sacandaga. So I'm going to cut to the chase and give you, I'll tell you kind of a little bit of the story at the end. So first off, overall, because this test was done in several different regions across New York, the great news that I can say is that we have one of the lowest invaded regions in New York. When our tests come back, we don't get a lot of stuff. That's a good thing. Sometimes you feel kind of bad to spend thousands of dollars and they come back and tell you, we didn't find very much, but that's actually a good situation to have. So they did this in the lower Hudson, they did this in Western New York, they did this in the Finger Lakes. We are the lowest out of all of those regions. That is because of all the great prevention, communication, our lake association, all of yourself. So we should be very proud of that. We only detected one new species to the region, and I'm going to put an asterisk behind it. We did get one positive sample of quagga mussel on Great Sacandaga. We need to do some further research and kind of looking into it, but it is kind of a, we did find one new species to the region. And as you'll see, there's really a lot of nuance with this monitoring method. And so we're going to kind of go through some of that. So there's things about it that work really well. Some things about it that don't work so well. So don't put away your rates. You're still gonna need them. Okay, another group of caveats. Some AIS species markers, especially for the plants, they can be positive for the native species. So they're looking at these really common genes. So they're looking at like chloroplast genes, or they're looking at these markers. So we talk about invasive milfoils. A lot of people in our comment, they're surprised when I tell them, well, we have 6 native species of milfoils. They're in the same Myriophyllum genus. They're closely related. Same thing with our Potamogeton crispus, curly pondweed. We have the most common plants we have in our lakes are Potamogetons, our native pondweeds. When we are doing these tests, most likely it's also detecting positive for those native congenetic species. Some of the low copy counts, when they do this, so Ampersand, they'll tell us the number of target copies they found in there. When they are really low, that probably could indicate a bias in the laboratory method. So the higher the number is, the more confidence we have, the lower it is, the less confidence we have. And once again, the test is just telling you that the genetic presence has been found in this sample. That means there could be a living organism in there. There could be a living population. It could be one single individual that got dropped in there. There could be a boat that came from Lake Champlain, which has zebra mussels everywhere. It has the eDNA of the zebra mussel on the boat. It does not have living zebra mussels on the boat. It goes to a new lake those environmental cells, eDNA comes off, we just happen to collect it. A bird ate a round goby in the Hudson River and flew over Schroon Lake and took a poop. We happen to collect that DNA of it. So there's always, the first thing is when you get a positive eDNA thing, the next step is always, we need to test again and we need to see if we can confirm it with another method. So overall, out of all those 89 samples that could have had 54 individual species each, we only had 33 positives. Very, very small, less than 0.00001%. Out of those, only 8 of them have more than 2 target copies on them. So once again, all of these other ones, especially when they're ones, that's at the very lowest level possible to detect. Kind of take those with a grain of salt. Not so sure how strong, it's not a very strong signal with them. Okay, so this is the name of the lake. This is the species and the marker that they use for it, and then this is the number of targets. So some of these a little bit above 3, we're starting to feel a little bit better on, but overall very low. So another way to look at that, out of the 89 samples, 27 of them had positive samples from 14 lakes. The average positive samples per lake was 35%. Okay, so tested 89 samples, we only had 27 of them come back. We detected 5 species: Asian clam, curly leaf pondweed, Eurasian water milfoil, quagga mussel, and zebra mussel on those. Here were our lakes that had positive eDNA results. This is the name of the lake. This is the number of samples we did. This is the number of positive samples for it. So Floodwood Pond in upper Saranac Lake region, 4 samples, 4 positive. So 100% of the samples had a positive in it. Lake George, 4 for 4. Tupper Lake, 4 samples, only 1 of them was positive. Loon Lake, 2 samples, 1 of them is positive.
Floodwood Pond, I grew up as a kid, that's so remote, surprised I had some.
Yep, well, we will talk about that in more detail. These are the lakes that had no positives on them. Interesting, out of these lakes, Friends Lake is the only one of these that we do not know. We have no known invasive species. So Fish Creek Pond has invasive species, Forest Lake has invasive species, Peck Lake. We even have a IMAP record of curly leaf pondweed that I probably don't feel too confident in your lake, but Upper Chattagate, Upper Saranac Lake, we know there's invasive species in there. No detections on these places. So I'm going to go through 4 of the 5 species, and I'm going to talk about the same types of charts in them right here. So the first one, this bar graph on the left, is Asian clam. This is kind of like our star, shows hey, this test is working well. Out of the 22 lakes that we tested, one of those lakes was known to have Asian clam, Lake George. Well, we detected it on Lake George. So that means we had 100% detection rate on there. So good job. The test is working how we want it to work. Zebra mussel. We have 1 known lake out of the 21, also Lake George, on there where we know it is. We detected it on 9 different lakes. Well, we did do it at 100% of the lakes, but we found it at 8 other lakes that we have no known adult living populations in. Lots of different discussion we can start to go into about them. The majority of these were very low target copies. Curly leaf pondweed. We tested 9 lakes that had it, and we found it at 9 lakes. But we found it at only 2/3. 6 of the lakes were ones that we already knew it was at. 3 of the lakes we didn't detect it on. So we found it on 3 different lakes that have no record of having curly leaf pond. Most likely, this is probably due to the native. Most likely, I personally believe all 9 of these positives are from native ones. If you think about it, we were testing in August and September. Curly leaf pondweed is the nest. It is sitting quiet and dormant on there, and it goes through that way. So Eurasian water milfoil. It is on 13 of the 22 lakes. So we go through there. We only detected it on 3 lakes out of there. And 77% of lakes that have it, we did not detect it on. So let's look at this on some maps. So here's Slowwood Pond. We did 4 things right here. We detected both native Potamogenians and Eurasian water milfoil. Here's Eurasian water milfoil map right next to it. So good, it worked. Here's Lake George. These are all Asian clams right through there. Oh, we detected Asian clams right in there. Yes, the system is working how we want it to work. Here's bad. Here's the boat launch in the village of Scroon Lake. 5 different samples. We know that there's Eurasian water milfoil in these blobs in there. No single positive on that. We did 18, 19 ones all throughout there. Not a single positive of Eurasian water milfoil in there. Here's here's Lower Saranac Lake. Here's 4 tests right here. 200 feet away is a thing of Eurasian water milfoil. Eurasian water milfoil in these different places on this bay, no detection for it. So, question, did we detect new species? Yes. But the new species on a water body, most likely the curly pondweed is from natives, and we have uncertain about the origin of the zebra mussel eDNA. We did detect quagga mussel in Great Sacandaga, and we'll need to follow up and do some more looking at that. Can we detect known invasives? Yes, we did detect known invasives in lakes, but not every time. Our lakes varied from 100% to 0% detection. The average was 19%. So, and the distance from the known AIS seems to be very limited. So, in summary, there's positive things about it. It's fast and easy to collect. The methodology works. We had no field contamination when we did these. The negatives, single sample is probably not enough from a water body. We weren't able to detect EWM on Scroon Lake. We'll be following up about that. And we have some uncertainty about these positive samples. So what are we going to build on this? In 2024, we're going to do fewer lakes, but we're going to increase the sampling effort. We're going to do 4 times more volume. We're going to do 4 times the number of location, and we're using a different filter. We're trying to catch more fish. Essentially, is what you're doing with DNA. You're trying to catch these genetic things. So we're increasing the size of our net through there. And we'll be doing it at multiple different locations through there. And we're going to compare this to the same 10 lakes that the early detection team is doing. So big thanks to Dana Holmland. She was our AIS assistant who collected this. The majority of these, big shout out to East Shore Scroon Lake and Scroon Lake Association who paid for some of these samples and went out and did some of the sampling. You can come find me sometime later and I'll be able to talk about any of this in more detail.
Totally awesome. I love the fact that we have a lot more to learn, right? Because that's what makes it interesting. And Brian has done a great job of thinking about the design for this summer. How do we test some of the things we learned? So we're going to actually switch gears pretty quickly. And Nancy, we're going to pull up your presentation now. And I loved these partner meetings because we also get a chance to hear from our partners. All of you are sitting on just as much information as the 4 APIPP staff who just presented. So I do hope you will stay for the lunch and the roundtable. But at this point, I'm going to call up Nancy Budd from the Adirondack Garden Club, who's going to talk with us about a great project they're doing with Champlain Area Trails and with the Adirondack Botanical Society. And as I said, you know, it really all works because all of us are doing different pieces here. So the Adirondack Park Invasive Plant Program staff, we're covering 6 million acres. We can't get down literally to too many pieces of the ground and actually think about what to do about a particularly important place. And that's where groups like the Adirondack Garden Club, being able to work with Champlain Area Trails and others, can actually make a big difference on the ground. So with that, Nancy, thank you.
So I am the project coordinator for the Aquatic Garden Club's invasive plant project at the Essex Quarry Preserve. And my contact information is, is down here at the bottom. First, before I even begin, I'd like to thank APIPP for inviting us to this partners meeting. It is a great honor for us, and we very much appreciate the ability, the opportunity to present our project. We welcome any suggestions that any of you may have regarding our project. So my talk has five parts. First, background information. Second, the invasive plants. 3, the infestation. 4, restoring native plants, and 5, future plans. So first, background information. Our project takes place on in the Essex Quarry Nature Preserve, which is public land, managed by Champlain Area Trails, and it opened to the public in 2021. The entire preserve is 35 acres, and it surrounds a small limestone quarry. The type of ecological community is limestone woodland forest, which transitions from white cedar along the lake as we move westward to a mix of white pine, red cedar, maple, oak, ash on the western side of the preserve. The forest floor contains abundant native wildflowers, grasses, and sedges, ferns, and mosses. And we found one endangered species, that's the state threatened handsome sage, Carex famosa. And I should mention that I'm a paleontologist, so occasionally you'll see pictures of fossils. Okay.
Millions of years old.
What's that?
How many millions of years old?
Oh, 450.
So the question for those on Zoom is how old are some of those fossils? And the answer was 450 million?
Yeah.
Million years old.
Okay, so these are satellite images of the Quarry Preserve, and this one was taken in 1952. This is 1986, and this is 2021. And I've put cues here indicating where the quarry is. The boundaries of the preserve are here, and here, and here. I actually put blue boxes around the boundaries so you can see it better. What you can see is in 1952, there was pasture and farmland to the north, and to the south, there was some woodland. And as we moved in 1986 and 2021, the preserve has become overrun with invasive trees and shrubs, so that today, the entire preserve is covered with invasive trees and shrubs, and the infestation is severe. We are working in a 1 acre area just to the west of the main quarry. And this is a blow up of that circle I just showed you. We have 4 different sites. They each have a different light regime and amount of moisture. Here again is the main quarry, site 1, 2, 3, 4. I'll tell you more about these sites. Okay, our project is part of the Garden Club of America's Partners for Plants program, and this program has a number of requirements. First, the project must be on public land. Volunteers must do all the work. We can hire out. Third, a botanical expert must provide us with advice. We're all amateurs, so we need advisors. And this last bullet is not necessarily a strict requirement, but it's a principle that we've been following. To every extent possible, plant debris is not moved away from the area, and no new plants are brought in from elsewhere. Okay, our crew of volunteers are all amateurs. Actually, all of us are all amateurs, and we're all volunteers. Our project began July 1, 2023. And in the first 6 months, we had 30 different volunteers working on our project. We average about 5 to 6 volunteers per weekly work session on the project. You can see we're working. One of our volunteers, Tim Rowland, actually wrote an article for Adirondack Explorer about his experience volunteering for us. He entitled his article "Dispatches from a Buckthorn Assassin." And a few of ours is a team project, and a few members of our team are here today. There's Laura Sales Doyle here, and there is Belinda Buff there, and Anna Moore in the back, and myself. And here in the background is Tony Feldman, and he's been helping us a lot with cats. And we also like to thank cats for allowing us to do the project on their land. And Steve Young and Carla Dornan are our botanical experts. They're over here. Steve, Laura. Okay, part 2, invasive plants. Our focus is on 5 invasive woody shrubs and vines. All were brought to this country in the 1800s. All are reported to be common in disturbed areas. All produce prolific fruits, for the most part, berries, that are dispersed by birds and other wildlife. Most leaf out early in the spring and shade other plants. You'll see that already happening with honeysuckle out of the quarry. And most of them shed their leaves late in the fall. And the seeds remain viable in the soil for many years. I should say many, many years. We don't completely understand how long they remain viable at this point. 2 of the most abundant invasives at the quarry include common buckthorn, Rhamnus cathartica, and various bush honeysuckles, Lonicera species. The buckthorn consists of woody shrubs and trees, which extend up to as much as 30 feet high and reach up into the canopy. So you see all these buckthorn trees. The honeysuckles are woody understory shrubs, and they don't get much higher than 12 feet. Most of them are much smaller than that. Both plants grow and spread rapidly, shading out native plants and depleting the soil of nutrients. They also release their allopathic chemicals into the soil. And these chemicals affect the soil chemistry, the nutrient cycling, the soil fauna and microbes, and they have a legacy effect. So that even after buckthorn and honeysuckle were removed, they still continue to inhibit the growth and even establishment of native plants. So many of our volunteers cannot identify buckthorn and honeysuckle, so we have to provide aids and identification aids to help them. And these are some of the characteristics that we use to identify buckthorn and honeysuckle. Buckthorn alternate leaves with finely toothed margins, blackberries. Honeysuckle is opposite stems and leaves with smooth margins. Red berries, you probably know all this. Distinguishing species of honeysuckle is particularly difficult because there are many different species. 9 species have been reported in the state of New York. 6 of them are native. 3 of them are invasive. And one way to distinguish species is to use the shape of the leaves, which is shown here, but this doesn't always work all the time. So what we tell our volunteers is that invasive species have hollow stems, whereas native species have solid stems, and we don't really bother, at least for our volunteers, with going any further than that. Here are 2 of the invasive honeysuckles that we identified so far at the Quarry Preserve. There may be others. One is Lonicera xyloscium, and the other is Lonicera moroi, and both of them have very similar shaped leaves. So the way to distinguish these 2 is by looking at the winter buds. The xyloscium has long winter buds, whereas the moroi does not. And another invasive that we've been tracking is particularly problematic. This is this Asian bittersweet, Solastris orbiculatus. This is a woody vine that wraps around and strangles tree trunks. It also covers the understory vegetation and essentially smothers them. And it has red to yellow, as it's shown here. The 4th species that we've been following is Japanese barberry. This is a thorny woody shrub with lots of small leaves. Here you can see it. Right now, there's only a patchy distribution of barberry at the Quarry Preserve, but we're watching it so that it doesn't become more abundant. And finally, the 5th invasive that we're working with is multiflora rose. There are a few of them. There's not a lot of them yet at the quarry. And this is a shrub with long arching canes. This is the arching cane. These are all maple seedlings. But anyway, long arching canes and compound leaves. So those are the 5 invasives that we've been focusing on. Now I want to tell you about the infestation. It is horrible, severe. Here again are our 4 sites. I showed you this before. 1 we call the entrance, 2 we call Stonehenge, 3 Midwoods, and 4 is Pond. And actually, I noticed that there's lots of water people here. We need to understand better what's going on in our pond. Okay.
And Nancy, just a note that the speaker is here for the folks on Zoom. So-
Okay, can people hear me? I have a very weak voice, so I hope that the group can hear what I'm saying.
Yes, I think so, but I think if you're turning towards the screen, it's a little bit harder. So you can also see what's up on the screen here.
Okay. Okay, so site number 1, this includes both part sun and part shade, and the dominant tree at this site is red cedar. Site number 2 has deep shade, and the dominant tree is white pine. For site 3, there's both a sunny area and a wooded area, and here the woods consist of a mix of different trees. It includes both white pine, red cedar, and a variety of different deciduous trees, oak, maple, ash, and so on. And again, the same, more or less the same mixture here at site 4, which is the pond. And here's a picture of this pond. And we have lots of questions about what's going on in the pond. So hopefully people here can help us with that when we go out and look at these sites. Okay, as we've been progressing, we've been entering information into IMAP invasives. And there was quite a big learning curve for IMAP invasives, but I think we finally conquered that. We used survey 1, 2, 3 to draw polygons around our 4 sites. And then we have been entering information directly into IMAP invasives. Here is some information that we entered for site 1. This is the entrance. This is before treatment. Here's a page for common buckthorn. Here's a page for honeysuckle. And the thing that we're most concerned about is the percent cover. It was 50 to 75% cover of buckthorn when we first started, and 5 to 25% cover of honeysuckle when we first started. And here's a slide summarizing percent cover at the 4 sites. Here you can see 50 to 75% cover at site 1 and site 2, and more than 75% cover at site 3 and 4, and honeysuckle 5 to 25 at site 1, and site 4, and 25 to 50% at 2 and 3. These estimates were made subjectively, and we would like to use more quantitative approach to this. And Champlain Area Trails has helped us with this. We are beginning to use transect information to better estimate our percent cover. So after eradication, these invasives will grow back, so our sites need to be monitored for many years. I should say many, many years. Okay, so the type of treatments we've been using are non-chemical. In other words, we don't use herbicides. There are 3 reasons for this. 1 is we don't have the licensing or the training to do that. 2, we don't understand how herbicides affect the soil. And 3, we don't have the financing to hire professionals. So everything we're doing is non-chemical. For buckthorn, we've been sawing the trees and then black bagging the stumps. The black bags are called buckthorn baggies. Here's a picture of a baggie. And essentially you put a zip tie around the bottom of it and then ties it onto the stump. The smaller seedlings we pull with a weed wrench, erratic, or by hand. We also pull honeysuckle and bittersweet with a weed wrench or by hand. And for barberry and multiflora rose, there's not quite so much of that. So we've just been so far hand pulled. And these are videos of one of our work sections. The one on the left is cutting buckthorn and piling debris. You don't need to hear what they're saying. It's mostly gossip and chatter. So that, the second video is covering stumps with buckthorn baggies. This is one of the interns from Champlain Area Trails. Covering the stump and applying the zip tie. Okay, so here are some before and after pictures. Here before treatment, after treatment, before treatment, after treatment. You can see there's a lot of barren ground that's left over, and somehow we've got to cover that with native plants, or else the invasives are just going to come right back. Okay, here are some panoramas that Marcel Doyle created. One is for site 3. You can see some buckthorn baggies over here. And here is one for site 4 and some buckthorn baggies over here. So we began our project last July. And since we began, we've cut down over 300 buckthorn trees and applied buckthorn baggies. So that's where we currently are with it. And of course, we create lots of debris piles. Some of them are big, some of them are small. The big debris piles we intend to burn on site. And we intended to do this last winter. We waited for a snowy day to do it, and the weather just didn't cooperate. We didn't have a snowy day last February. I don't know if you remember that. And so we'll try again next year to burn the larger piles. The smaller piles we've been designing to be wildlife habitat piles. So in a wildlife habitat pile, the sticks at the bottom are large, and then they gradually get smaller as you go up. And there's a crisscross pattern to designing the habitat pile. We've seen different types of songbirds fly out of the piles that we've created. And we feel that once we've got the invasives removed, that the preserve will prove a great place to go bird watching. So, of course, we've been entering our treatment information into IMAP invasives, and this is for site 3. And our treatment goal is eradication, probably overly optimistic. And then this is a bunch of information that I've just gone through with you. Okay, part 4 of the talk. Restoring native plants. This is probably our biggest challenge. Last fall, we collected 52 bags of seed from 25 wildflower species. These include Allegheny monkey flower, which is here, the purple thing, partridge berry, baneberry, thimbleweed, various types of goldenrods, many different types of asters. And we intend to continue to collect seeds this coming fall, but also for spring blooming plants as well. And we also would like to start propagating grasses and ferns and sedges. Okay. As we collected the seed, we took a picture and we entered the pictures into iNaturalist. Our project in iNaturalist is called Essex Quarry Biodiversity Project. And rather than just direct sowing the seed, we decided to do something a little more controlled. And last January, we sowed the seeds into pots, flats, and milk jugs. And we left them out in the snow to cold stratify. And here you see Melinda entering information into our seed database. We have a seed database. And I'm pleased to say that as of earlier this week, more than 2/3 of our milk jugs and flats and pots have germinated. And so our next step is to keep these things alive and plant them next fall. We have been taking cuttings. There are not a lot of invasive shrubs, or I should say native shrubs at the quarry. But we did manage to take cuttings of elderberry, willow, I think I can remember, dogwood, and balsam poplar last March, and we just recently planted them. So part 5, what's next? We have funding for a 3-year project, and we're currently beginning year 2. During year 2, we'll do more or less the same sorts of things as year 1. We'll continue working at the same 4 sites. We haven't finished them by a long shot. We'll focus on removing invasive shrubs before we were focusing on invasive trees. We'll check buckthorn baggies applied during year 1 and cut any shoots. We anticipate lots of shoots. We'll experiment with other non-chemical removal methods, including burning. We'll create wildlife habitat piles, and hopefully we'll be more successful at arranging to have large debris piles burned. And we'll restore native plants by propagating seeds and cuttings and planting them at the quarry. So these 2 pictures on the right show what happens when you don't put a buckthorn baggie on top of a stump. Shoots everywhere. We anticipate some of this. So future plans. Our project is only a pilot. It involves volunteers working in a limited area for a short length of time. We hope that our project will be used to develop a more comprehensive strategy to restore the ecosystem through the entire quarry preserve, that is all 35 acres as opposed to just 1 acre, over the long term. We also strive to inform the public about the threats that invasive plants pose to our woodlands across the Champlain Valley and beyond. And we have lots of people to thank, organizations, APIPP, Champlain Area Trails, Garden Club of America. Mitchell O'Neill helped me a whole lot with IMAP invasives. It took me a while to figure out how to make it work. We have our botany experts, Steve Young and Laura Leitonen, and all of our many volunteers.